So let's focus just on the liver. Here is the liver. This horizontal section shown on the left here is actually viewed from the bottom up. So that's why the liver is on this side. On you, that's like over here, and we're looking from the bottom. You can really see how large that liver is. This says anatomy here, but I'm also going to do, of course, and some function. Do that first, and then we'll look at this picture and another view that shows the circulation to and from the liver. So the liver, you already know, it's going to produce bile. That's going to be what we focus on here. Those hepatocytes in the liver make bile. It's also going to store glycogen. So glycogen is a polysaccharide of glucose that's going to be broken down when needed with stimulation from the pancreas endocrine cells, right? Glucagon will break down glycogen. It's also going to store other nutrients and stuff, other stuff, good stuff, nutrients. Also important for like detox, dealing with drugs, alcohol. So we'll briefly touch on that. The main thing, again, we'll focus on is the hepatocytes producing bile. We'll focus actually not on how they produce it, but what that bile does. Okay, this view here is the liver, really big thing. The liver itself, I'll show you a picture of this in a bit, has these things called portal triads. Actually, why don't we wait and use that word when we get there. This here, though, there's the reason triad came up. There are three key things here that you'll look at. Artery, it's just the artery, right? Oxygenated blood coming from the aorta of the heart originally. Portal vein, that's actually an incoming vein. A portal vein is different than a vein. It's this portal system, kind of like your hypothalamic portal system, local, deliberately shutted from the digestive system to the liver. We'll look at this again in the next slide. Different from the portal vein is the hepatic vein. This is what's going to go to the heart. This is pretty much fully deoxygenated. This incoming vein here is only partially deoxygenated. The last piece of this triad here is the hepatic duct, right? That is going to carry bile to the small intestine and or gallbladder. So let's look at this piece, this hepatics, this circulation with a better picture. Here it is. Okay, could you label things on this picture? Start labeling, see if you can do it. Right? What else would you know right away? We've got an artery going to the liver. That's going to be your hepatic artery. Remember, hepatic refers to the liver. That guy? Inferior vena cava, coming from the lower half of the body, is going to be deoxygenated blood. So then what does that mean this thing is here? This has got to be our hepatic what? Vein, right? These are the capillaries. That allow for exchange at the liver. Come back to that in a moment. Oxygen and carbon dioxide are going to be two things that are exchanged. I have here labeled digestive tract arteries. So you probably could guess this is going to be our capillaries of the digestive tract. What is exchanged in the capillaries of the digestive tract? Oxygen, carbon dioxide. So here we're going to have O2, CO2, right? But what else? We're going to have absorption of nutrients. So nutrients are going to be absorbed from the digestive tract. Nutrients are now in this thing right here. What is this? Our hepatic portal vein. Here it's blue, but this is actually partially deoxygenated. So actually we're going to provide some oxygen to the liver through this system as well. But I don't care if you know that, because you've also got your hepatic artery, right? The liver needs oxygen. So this delivery of these absorbed materials directly to the liver highlights the importance of this organ as a biological filter, right? We've got nutrients and toxins, drugs, any drug you take. So the hepatocytes are going to contain enzymes that metabolize drugs, help clear toxins from the body before what? They go into systemic circulation. Because as soon as you go out into here, lungs, the aorta, you can't control where this is going. You want to deal with those toxins as soon as they come into the body, not just like anytime, as much as possible. And this is actually a reason why with drug doses, if you take an oral form, you have to take a higher dose than if you do a intravenous, right? Because intravenous is going directly into your bloodstream, not directly to the liver first. Pretty cool. Okay, we're going to zoom in to a little section of the liver. You're going to see this more closely in lab. We're not going to do a ton of detail on this, even in lab. Okay, so zooming into a section of the liver, you've got these hexagonal structures here. If you zoom into one of them, each of them has this central piece and then is surrounded by some key structures. And these surrounding structures on the outside here, this is actually these triads. So triad is like 3, right? That's going to be, you can't see them on this picture, but they're like out here somewhere. Central vein, we'll come back to. So these triads, here's one of them labeled. Each one's made up of the bile duct, the portal vein branch, and the hepatic artery branch, so that every section of this liver receives oxygen, receives nutrients and toxins that need to be uptaken or been dealt with, and is able to put bile into the bile duct system. So these hepatocytes all along here are producing bile. That's what that green stuff is in there. That's all going to be that's all going to be shunted to this bile duct and then meet up. Here's the kind of meet up to form this main bile duct. We'll see this again in a moment, actually. Point here is that this triad has all 3 things the hepatocytes need to have near them: a place to put the bile, a place to receive oxygen, a place to receive in that portal system, the nutrients, toxins, everything that's coming from the digestive tract. In the center of each of these hexagons is the central vein, where all of the deoxygenated blood is going to end up from this whole system and go to the hepatic vein, then to the inferior vena cava. So bile produced in these hepatocytes and then shunted through these little canaliculi to each bile duct of the portal triad. These portal triads, if you can imagine, okay, let's draw both sides, are going to meet up and become, let me show one more over here, become what? This is a lovely picture here of my bile duct, right? The common bile duct that's going to go to the small intestine. That's all you need to know. So small intestine backs up to that gallbladder when there's too much. Actually, this is a great color here for talking about bile. So produced in hepatocytes. What is it? It is a yellowish, brown, green, alkaline solution, substance solution. So solution, it actually is in water. It's going to have bile salts. That's an important one. It's going to have some other fats in there, phospholipids. Do I want cholesterol, triglycerides? I'm just going to write down the most important ones here. Bile pigments. That's what gives it its color. One of these is bilirubin. I think I spelled that right. Yeah. That's the main components. Electrolytes. I'm actually not going to talk about those, so I won't write it. These two here and bile salts are going to be what we talk about the most. Bilirubin, just as a little bit of a side note, it is produced in the spleen due to breakdown of red blood cells. The spleen's job is to recycle red blood cells that are old or damaged. Breakdown of red blood cells produces, among other things, bilirubin. Bilirubin is actually a toxin that is going to be, by putting it in the bile, we can get rid of it. We'll see that in a moment, but this should make sense, right? We are putting bile into the small intestine. The small intestine is going to enter the large intestine and then enter, produce feces. So it's a way of getting rid of this toxic substance. It's not the only one, but it's a main one. And maybe so high and low bilirubin, well, high is a clinically relevant thing to look at. Okay, so let's talk about bile salts. Pretty cool stuff. This is a globule of fat, as I have labeled. Fat is what? Hydrophobic. So if we want to be able to break down fat, ultimately by an enzyme, we need to be able to break this thing up. This is called emulsification. It's actually a type of mechanical digestion where we're disrupting those hydrophobic interactions. How do you disrupt hydrophobic interactions? If you have grease on your hands or like oil on your hands, how do you disrupt those hydrophobic interactions? You use soap, a detergent. That's basically what bile salts are. They are just like detergents. They have a hydrophobic and a hydrophilic side. What's this called? Amphipathic. Just like your phospholipids. So phospholipids actually can act like this as well. Bile salts and phospholipids are both present in your bile. Bile salts are more prevalent. So because this hydrophobic side can interact with fat, it can get up in here and break apart these large globules. That's what's shown here. It stabilizes fat in smaller globules. That's called emulsification. That's it. And that's what happens when you wash soap off your hands. So this is smaller fat globules now. Let's write here a large fat globule. Why do we want to do this? Well, this is still like a whole clump of like triglycerides. We still need to be able to chemically digest that, but now we can do that. So this is a little preview into absorption here that kind of shows this here, same large fat globule we're starting with that's you ingested some delicious fat. Bile salts are going to emulsify. Then we can actually, here, this is a change in chemical structure here. So this is chemical digestion. What does that? This is going to be a lipase. And this is actually, let me make sure I have that right. Yes, I have that right. A pancreatic lipase. Lipase breaks the bonds to go from like triglycerides to fatty acids. These are called micelles. Not sure if I'll come back to that. They're basically stabilized by bile salts again, so that they can be carried and absorbed into these enterocytes. How can these get through here? What's going on there? Well, they're hydrophobic, so they can just go right in there. When we get to carbohydrates and proteins, it won't be so complicated. They can diffuse through the phospholipid bilayer and go into the lacteal, where they're carried away into the lymph. And that's what's unique about fats.